And this is the HPLC analysis of the extract of that tree bark. And as you can see, the extract contains many different compounds, and 4 components are main components. As you can see, these four peaks. Those compounds are QS7, 17, 18, and QS21. Structures of those compounds are shown here on this slide. As you can see, they differ in a structure on those sugar units. For example, those 3, they have this complicated chiral acyl chain. This QS7 has just a small acetyl group here. The most important representative from this group of compound is QS21. It is one of the most potent adjuvants known to date, which after over 100 clinical trials, it was finally approved as a component of GlaxoSmithKline adjuvant formulation for human vaccine Shingrix. It induces a balanced Th1, Th2 immune response, and actually when we develop new adjuvants, we want that from some new adjuvant molecule. But this immune response depends on the acylation of the fucose unit. This is fucose sugar. And if this fucose bears this chiral acyl chain here, then we have mixed immune response. But if this chain is cleaved from this molecule, so if we have just OH group, then we have just Th2 response. This compound is not very stable. In water, this is prone to hydrolysis, so we will get just the acylated compound after some time. So this compound has major drawbacks in chemical instability, in limited supply, and very difficult and low yielding purification. And also, as you can see, this compound is extremely complex in the structure, and the aim of all groups who do the research in this field is to simplify the structure of Quillia saponins to make them more easily accessible. Marciani and his group did something very important in that way. They found a GPI adjuvant. So they used just commercially available raw tree bark extract, and they put it in reaction conditions to hydrolyze everything that was bounded here. And after that, they installed this alkyl chain on this side. In that way, by installing this alkyl chain here, they managed to retain the mixed immune response. But this adjuvant GPI is actually a mixture of many different compounds in which some are structurally defined, like those 4, but there is many other structurally unknown QS saponins. And for human use, that is not good, because we don't know the activity of all compounds which are present in the GPI. So some compounds can be even toxic. And we were interested to identify the active compound, structurally defined compound from GPI, which is responsible for its immunoadjuvant activity. As you can see here, the most abundant saponins in raw bark extract are QS17 and 18. And our goal was to synthesize structurally defined compound, which is QS17-18 analog. And this structure is shown here on the slide. And after synthesis, we evaluated the immunoadjuvant activity of this compound. So this is the main component in GPI, and it is structurally defined. Now I will talk about chemistry, because this is the main part that I do. So when we have some structure complex like this, we try to reduce the complexity in every step. So in the first step, we can disconnect this alkyl chain from here, and in the next step, we can break this bond here
to get this branched oligosaccharide and this compound here, which are both of them are shown on this slide. Now, when we have those two molecules which have a similar complexity, we can develop their synthesis. For branched pentasaccharide, the synthesis was done from monosaccharide units, but for this compound, the synthesis was actually much more easier. Why? Because tree extract contains many different compounds, and this is a common part in all of those compounds. And after hydrolysis of all of those compounds, we obtained this compound 1 in quite a decent yield of 50% mass yield. After that, we did some chemistry on this compound, installed protective groups and leave this carboxylic acid free in order to react with our pentasaccharide. For this oligosaccharide synthesis, we used two-stage allyl glycoside donor activation approach, which was developed by Professor Wang, with whom I did my postdoc in the United States. So I will just show briefly the main principle of this process. So first we installed a allyl group here on the sugar, which serves as a protective group. Then after switching this double bond to this position, this molecule is activated for the reaction. And after activation, it can react with another sugar molecule to give this disaccharide. As you can see on this disaccharide, we still have this allyl group, which origins from this sugar. And we can repeat all this process as many times we needed to synthesize our pentasaccharides. So the main part of in this work was the preparation of appropriately substituted monosaccharide units. And after 11 steps in total yield of 4%, we obtained our pentasaccharide molecule. After activation of this pentasaccharide, we attach it to the quinoic acid, to this part, and we got a compound. We got our final compound, which still has protective groups on OH groups. So next step was global deprotection, so removal of all of those protective groups and formation of amide on this side to install this alkyl chain. And that way we obtained our QS1718 analog. And we had a compound for immunoadjuvant activity testing. But I must also say that in this reaction with hydrogen over palladium, we obtained also a different result with different batch of this catalyst. We were actually very lucky because this different batch catalyzed reaction in a different way and produced this compound, which has all OH groups free, except those two on the fucus. Those are acylated OH groups. And we concluded that this is our QS7 analog. So from one synthesis, we got the opportunity to test two different compounds, which are quite abundant in the GPI adjuvant. And here is the evaluation of the immunoadjuvant activity of those compounds. We evaluate the activity in mice model using two different antigens. RHB is an etiologic agent of periodontal disease, and DNAK is a recombinant heat shock protein. We did subcutaneous immunization with in three groups of mice. One group was immunized with just antigen, another group with antigen plus our synthesized analog, and the third group was immunized with antigen plus standard compound, which was GPI or QS21. And serum was analyzed for antibodies after 2, 4, and 6 weeks.
Here we have the results. So our QS1718 analog, without an adjuvant, antigen alone produces very weak immune response, as you can see here on this graph. GPI produces quite strong immune response, much higher antibody count, and our compound is here in antibody count in higher dose and in lower dose. And as you can see, this activity is quite comparable to the activity of GPI. That way we proved that our compound is probably the responsible compound for the activity of GPI. And another thing is that our compound can be structurally defined alternative to the GPI. We were also interested in the profile of the immune response. So we found that our compound shows mixed Th1, Th2 immune response. It produces both types of antibodies. For our QS7 analog, we found that it produces also strong immune responses, which are comparable to the immune response of the GPI adjuvant, even in lower doses. And we were also interested to test compound which has AC group here, which is acetylated in ester form, and compound which has OH group here, so hydrolyzed ester. And our hydrolyzed ester showed much less activity in Th1, so our acetylated compound showed mixed and balanced Th1, Th2 immune response, while compound with OH groups here showed immune response which is more Th2 oriented. After that, we were interested in detailed studies about the structure-activity relationship on activity of QS7 analogs. So we decided to synthesize compounds which have this oligosaccharide domain truncated to smaller oligosaccharides. And our goal was to synthesize compounds. This is a common part of all of those compounds, and this is only part which is different. And for this R group, we attached just fucose and got monosaccharide, fucose and rhamnose and got disaccharide, trisaccharide, and tetrasaccharide with native configuration, exactly the same like in natural QS21, and different tetrasaccharides with configuration which is not native, so a natural configuration on this side, or with the carbonate ester on this side.